It's my pleasure to moderate this session, and I am Zohra Hossainzadeh, chair of this panel and chair of Life Science Selection of the Young Academic Europe. And today we would like to simulate an ERC starting interview, life science. Before I start the session, I would like to thank Academia Europaea Budapest Hub and Hungarian Academy of Science for organizing this events, and also Young Academic of Europe, and particularly Kathleen, I hope that I pronounced it correctly, Borvolgi, ERC National Contact Point, Nuradik, Academia Europe Budapest Hub Officer, and Aniko Dunitzti, Hungarian Academy of Science. So as I said that today we simulate the ERC interview and LS panel, LS2 panel. Today we will hear from the Dr. Nitzan Gonen, who received an ERC starting grant in 2021 under LS2 for project enhancing sex deciphering gene regulatory networks governing mammalian sex determination. And the panel are Dr. Gautam Dey from EMBL, and myself, and Professor Gaspar Jakawi, COS Heidelberg University, Professor Sebastian Leidel, University of Bern, Professor Mangala Srinivasa from Wageningen University, and Professor Shahrukh F. Shariat, Medical University of Vienna, and Professor Mark Yest, University of Girona. For today's session, Dr. Gonenan will begin with a 7 minutes presentation, and we will follow 18 minutes Q&A from panel. And I ask warmly the audience that submit the question in the chat, and if remaining time, we can also ask this question. And of course, after panel session, we will have a time to discuss some technical aspect. Please mute your microphone, deactivate cameras, and this also events, I should say, that record. So then, Dr. Gonen, I invite you to start. You have 7 minutes. I am a chair of the LS2, and we would like to you present your project today.
Thank you. Can I please share?
Yes, please.
I need you to, okay, great.
Yes.
Okay, so thank you so much for the invitation. My name is Nitzan Gonen, and I'm really thrilled to be here. So is it a boy or a girl? This is the question we all ask very often during pregnancy, and we do that because our sex, our biological sex, is a very important determinant in our life, and this is what we study in my lab, trying to understand how this process happens and how it is regulated. So during sex determination, a bipotential gonad, a shared organ, can differentiate into either testis and ovary or an ovary. And while we know a lot of the genes involved in that process in the last 3 decades or so, it's striking that we still don't manage to explain many of the cases that present with sex reversal. They are called DSD patients. In 50 to 80% of the cases, we will perform exome sequencing, meaning sequencing all of the coding genes, and we'll still not be able to solve the problem. And that may be because we are only looking at the 2% of the genes that encode in the genome, and the answer may lie in the 98% of the genome that are non-coding. And in addition, while we don't know much about the non-coding genome, while we know those factors, we don't really understand how they all interact together and how they work together in a system. So why me to tackle this question? I've always been fascinated about gene expression regulation throughout my career. And 2 years ago, I established my lab at Bar-Ilan University, and we're studying the process of sex determination. And most of my career and knowledge comes from my postdoc at the lab of Robin Lovell Batch at the Creek, where I've kind of laid down the concept that regulatory elements can be involved in sex determination and in DSD pathologies, where we've shown that deletion of a single remote regulatory elements of the SOX9 gene can lead to sex reversal in both mice and human, leading to a phenotype like mutating the gene itself. So I think that if we want to try to understand DSD cases and to better understand sex determination, we need to look at the big picture. And what do I mean by that? We know that sex determination is orchestrated by the joint activity of a lot of key pro-male factors and pro-female factors. And while we know what each of them does alone, we don't really understand how they exert their activity, what are the genes they regulate, and how they do that. So in that proposal, what I suggest is to identify all of the direct target genes of the key pro-male and pro-female factors. And in addition, I also want to understand how they exert their effect, what regulates them directly, very similar to what I've done for the SOX9 gene, but also how they regulate their direct target genes. So to tackle those questions, we need to overcome two major hurdles in the field. And one of them is that we need to be able to incorporate low cell number techniques, because the gonads develop at embryonic stages where the biological material is very limited. And in addition, we need to be able to purify specific cell populations in the gonad, because all of the activity of sex determination happens in Sertoli and granulosa cells, so subtypes of cells in the gonad. And a lot of those key markers are expressed in more than one cell type. So we need to be able to purify the cell type of interest. So in this ERC proposal, what I plan to do in AIM-1 is first to identify the target genes of the key pro-male and pro-female factors. We will do that using a new reporter mouse line that we've generated called Enhancer 8 and Cherry that enables us to very efficiently purify both Sertoli cells and granulosa cells from the ovary. And using those mice, we will perform cut and run ChIP-seq, a technique that is highly suitable for low input cell number. Roughly 10,000 cells can be used for cut and run. And we will try to identify the direct target genes of the pro-male factors and the pro-female factors. That will be integrated with RNA-seq, again generated on those cell types, and that will allow us to identify both the direct genes or target genes that are both activated and repressed by those key transcription factors. And together that will allow us to start building these gene regulatory networks that control the process of sex determination. In my second aim, I want to tackle a similar question, but from a different angle, and this time not looking necessarily on the key transcription factor that control the process, but rather on the regulatory elements that they exploit in order to execute their effect. To do that, we will use the same reporter mouse line and perform several genome-wide genomic approaches to identify, so including ATAC-seq, histone ChIP-seq, and promoter capture high-C. And by doing that, we will first identify all of the regulatory elements that participate in the process of sex determination. We will be able to classify them as enhancers, silencers, and insulators and promoters, and we will be able to direct them to their direct target gene. I assume that we will find numerous interesting candidates, and we will want to validate some of them in vivo using CRISPR genome editing in the transgenic facility that I set up. And we will choose the candidates to model based on whole genome sequencing data that we will get from whole genome, from DSD patients, from my collaborators. By doing that, I think we will first be able to better build this gene regulatory network, but also shed light into the importance of the non-coding genome in this process of sex determination. Some preliminary data that we have already from the lab is that we performed using those specific reporter mouse lines, ATAC-seq on E13.5 and E15.5 Sertoli and granulosa cells. And while we were able to identify previously known enhancer like enhancer 8 of the SOX9 gene, our mouse line also allowed us to identify novel enhancers that haven't been picked up before, and we believe that this is due to the more superior mouse line that we're using compared to the one used before. So AIMS 1 and 2 will give us multiple candidates, regulatory elements that may be involved in that process. And while we will be able to screen some in vivo, we will not be able to screen all of them in mice. Therefore, it's extremely important that we will have an in vitro system to model them and to study better the gonads. To that aim, in aim 3, what we plan to do is to develop testis organoids that will be generated from both primary testis samples, but also from stem cells that will be differentiated towards the somatic cells of the gonads. And I want to show you some preliminary data on testis organoids we were able to generate from primary testis. And you can see that they get organized into very similar structures like the real testis. So using that approach, we will be able to both study many of our candidate genes, but also having that system will enable us to study many open questions in the field of reproduction. Altogether, this approach, which takes a system biology view of the gonads, will help us identify or understand the gene regulatory networks of sex determination. But looking more broadly, I think that this system will open up a lot of other avenues of research, including our ability to hopefully improve the diagnosis rate and enable those patients to have prenatal screening. And with that, I will end, and I want to thank you.
Thank you so much for your nice presentation. I have myself a question, and also member of the panel have question. I may start with my question, and the member of the panel may hand up in the react, and then I will ask them to answer the question. One of my questions is why you are only this time point of the E30.5 chosen for epigenomics analyze. Why not earlier, like E10 or E11?
So this is a good question. So the gonads start to develop at E11.5. E12.5 is kind of the peak that everything happens, and by E13.5, the story is almost closed. So like you suggested in that proposal, we propose to do the analysis on multiple stages. Of course, the younger we go with the stages, it's becoming more limiting in terms of the size of the tissue and the number of cells we can retrieve. But at least for the attack and the histone ChIP-seq, we're planning to do that on E11.5, E12.5, E13.5, and E15.5 as a later time point to after the sex determination has already been completed. And for other techniques that are more challenging, we will start with E13.5, and depending on how it goes, we'll see if we can extend it further.
Mm-hmm. And if you move to the ChIP-seq, how will you address the issue of the low cell number, reducing sensitivity?
Yeah, so ChIP-seq is indeed a technique that requires millions of cells for good quality, and this is why many people haven't been able to do that in our field. But recently there have been developments in a technique called cut and run by Henikoff, which have been employed by many people in various fields. And this technique has been shown to be very feasible with as few as 10,000 cells, especially for histone marks. But even for transcription factors, it seems to work with low input. There are modifications in the technical ways the technique is being done that enables it to work as low input. And having that technique along with our superior mouse line that we generated, because the previous mouse lines we could get roughly 4 to 5% of the cells of the gonads were indeed Sertolian granulosa, and with our line between 18 to 40% of the cells are Sertolian granulosa sorted, so we get much more cells. We are able to capture all of the cells. So I think that the combination of the low input technique and our enhanced reporter mouse line now paved the way for us to be able to do it with low input.
And how will you assess the truly functionality beyond the gene expression? Because you are doing gene expression a lot, like protein modification, signal interaction, protein-protein interaction. So again, it's a very valid point. I mean, when you do multi-omics, you discover a lot of candidates, but then how do you drill down into the mechanism? So in addition to my lab, I've also set up here in my university a transgenic facility.
where we can do CRISPR genome editing for any target that we want. And of course, we're very interested at regulatory elements. So very similarly to what I've done in my postdoc, where I've deleted a regulatory element and I've shown that it's critical and a single regulatory element can phenocopy the phenotype of knocking out the gene. We're planning to do something similar with the candidate enhancers we will find from our screen. Once we will find the interesting candidates, again, choosing them based on them being regulatory elements for the key sex determination genes, and they are open in more than one stage, and we have more the histone marks and the attacks, so a lot of things that will make them more favorable over others. We plan to delete them in mice in vivo, and then both assess the gene expression of their target gene during gonad development in vivo, but also see if it can induce some kind of a gonadal or reproductive system phenotype.
Thank you very much. Now I would invite you other panelists to ask question. Yes, Dr. Dave.
Thanks very much. This is a very, very interesting presentation. So I had a couple of unrelated questions. So about your organoid testicular model. So can you explain what readouts you would use to verify that you've generated, something that is 80% of a functional gonad or functional in X or Y ways? Like, so how will you benchmark this, given that there is no precedent, right?
Yes,
It's like an exciting goal, but yeah.
So it's a good, valid question. So one of the reasons that we are kind of left behind in our field is that we don't have an in vitro system, and everything that you discover you need to do in vivo and generate mice takes 3 years and so it's very challenging to move forward. And like I said, because it all happens at embryonic stages, we're very limited with biological material. So having a model system that is in vitro, and even if it's not going to mimic 100% the real tissue, but it will be a good enough close proximity, which I assume organoids are as such, they are never 100% of the real tissue, that will be a major breakthrough for the field. So I've developed during my postdoc a differentiation protocol taking mouse embryonic stem cells towards the somatic cells of the gonad, mostly Sertoli cells. But then we realized that, okay, great, I generated very similar transcriptomic-wise cells to Sertoli cells, but how can I keep them in vitro for a prolonged period? And that raised the question of how can we culture them for a long period? So then instead of going to the stem cells and trying to find out how can I culture a testis in a dish, we said, let's go to the real tissue, to real testis, and try to dissociate them and see what 3D settings, what defined media we need to provide them in order to reorganize. So the plan is to first work with them and then assess their functionality by qPCR, by RNA-seq, by immunostaining, looking at the morphology, looking at the organization, looking if all of the cell types that normally are present in the testis are present in the organoids and being maintained, develop the media culture, the media compositions that enable to maintain them in vitro. And once we will do that, that will serve as a good platform to then replace the cells with stem cell derived cells, because I assume that not many males will want to donate their testes for research. So we eventually plan to do that from stem cells.
Thanks very much. And now just zooming out very much. So in general, given the speed of regulatory evolution, and especially in non-coding elements, so can you sort of talk a little bit about how you expect, let's say now your kind of systems biology level findings in mice to translate to other mammals?
Yes.
Is there any way to get a grasp on that?
Yeah. So you're very right. And as far as genes being conserved, regulatory elements are far less conserved. However, there are regulatory elements that are conserved. And for example, the enhancer that I discovered during my postdoc, enhancer 13, is conserved from human to mice and many other mammals. So I believe that the critical ones may be conserved. But in parallel to my work, I know that others have been trying to do, including the lab of Roser Ventotormo in the EMBL, not EMBL, in Cambridge. They have been doing single-cell ATAC-seq and RNA-seq on developing human gonads. So we will have from their data the open chromatin regions from the human, and we will have ours from the mice. And the plan is to later on intersect those two and to be able to identify regulatory elements that are unique to each and regulatory elements that will be conserved.
Thanks.
Doctor, yes?
Yes, thank you. Yes, thank you for the presentation. So just yes, as well, a couple or three questions that I have from the presentation. So my first question is that you mentioned about checking the data from patients using whole genome sequencing, and it was not clear to me because you mentioned that this came from your collaborators, and it was not clear to me how this selection of patients would be conducted.
Okay, thank you. Thank you for raising up for that. I'll be able to better explain that. So my lab doesn't work directly with DSD patients, but I have strong collaborations with two labs that are big hubs that collect to them a lot of DSD patients. So when those DSD patients arrive and they know to classify them to the type, whether it's primary gonad problem or whether it's a hormonal problem, meaning they couldn't generate the hormones, testosterone or estrogen, or whether they couldn't respond to that. We're mostly interested in the primary sex determination problems. So then what they usually do to those patients is that they do exome sequencing or gene panels on known genes to try to identify the mutations. The situation is that in 50% of the XY DSD and 80% of the XX DSD, they will not find anything in the exome. Even if they sequence all of the coding genes, the ones that we know that may be involved and the ones that are not, they do not find variants that could explain the DSD. So then what they do to those patients is that they perform on them whole genome sequencing. And then the idea is that we will get that information from whole genome sequencing and see whether we have a collection of 100, 200 DSD patients and there is kind of hotspot enhancer. So there is region that we found that we suspect may be important, and we see that there are many variants in DSD patients in that region that will highlight it as a more likely enhancer we will want to target in mice.
Okay. Thank you for the clarification. So another point that I also had is that so when thinking about organoids, so I think that now the main proposal is based on organoids for the testis, or also you are attempting to also provide a model for granulosa cells and the ovary. And if this was the case, that you plan that the future as a follow-up, how do you think that the nature of the Sertoli and the granulosa cell, the difference between these two cells, can make a difference in terms of having enough material to do the analysis, enough material to analyze the gene regulatory elements and so on?
So again, a good question. So actually, the field of ovary organoids, or what it's called ovaroid, has been much more progress than the testis organoids. And there is two groups in Japan, the Hayashi and Saito Mitinori group, that are extremely strong on that avenue. And they have been able to generate ovary organoids, which kind of form a follicle where you have an oocyte in the middle and you have granulosa cells around. In the beginning, they've done it with granulosa cells that they purified from primary cells, and they wrapped an oocyte that was made from stem cells, and they showed that they can fully mature them in vitro into functional oocyte. And they went a step further and injected the sperm inside, and they showed that they can generate pups and healthy and fertile pups. So we were able in mice to generate from stem cells a healthy pup. However, this has not been done in the male. I'm sure they are trying to do that, and I'm sure they may be my competitors. But I think that the testis has its own complexities. But what is similar, you were asking before, what will be similar and what will be different? I think that in both cases, the germ cells are, of course, the very important cells that contribute to the production of oocyte and sperm, but they must rely on support that they are getting from the somatic cells or the somatic niche. And in that respect, it's the Sertoli cells in the testis and granulosa cells in the ovary. So I think, and I'm more of a somatic cell person. So I think that once we will be able to generate the best somatic cells and the best niche media and 3D setting, then we will be able to, as much as possible, allow in vitro gametogenesis or a better modeling of the gonads in vitro.
Dr. Leide.
Okay, thank you. I have a question. I have no doubt that you're a brilliant person in the wet lab, and I understand that you're using very fancy methods and different ones which will generate tons of data. What I do not understand at all in your proposal is how do you analyze your data? You write something about computational experts at Bar Ilan, who I don't know how they know about all these different techniques. How will you select out of your 10,000s of data points all the loci that you actually want to modify?
So that's the
you can't
question.
do 1,000, I guess.
That's the question that I was most terrified about, because like you mentioned, I'm a wet biologist and I'm suggesting to do here a lot of multi-omics that requires high level of computational analysis. So true, I'm not computational, but doing those integration nowadays is not something that is very unique, and there are already ready-made pipelines for analysis of each method alone. So we have done already and analyzed ATAC-seq. We are very experienced at analyzing RNA-seq. The promoter capture Hi-C is being done in collaboration with labs that are expert in promoter capture Hi-C, so they will provide both the experimental and computational analysis. And I already have in my lab a highly trained computational biologist, a postdoc that is both very good computational postdoc, but also very expert in sex determination, who will be able to integrate all of those approaches. The idea is to basically look at the gene expression, look if genes are expressed or repressed, try to do linkage analysis, linking the open chromatin regions to the gene, both positive regulation, meaning that the enhancer is open and the gene is expressed, but also negative regulation, that the enhancer is open, but the gene is blocked. And those could be silencers. And then the idea is to look at the topological association domain, so TADS, so the gene borders or the gene regulatory borders, to try to assign an enhancer to its target gene. And then using our data from DSD patients or the data, like I suggested before, of conservation to human enhancers of the gonad, that will allow us to prioritize the candidate enhancer we will want to further model. And in addition, we will, of course, focus first on genes that we know that are critical for sex determination. And those are all of the genes that appeared in my first slide, all of the key transcription factors that control sex determination. So, of course, we will first start by looking on regulatory elements that regulate these genes.
Yeah, but then you are doing basically what everybody else does. What brings you further? That is what I don't understand.
So nowadays, no one has yet done everything I've suggested doing. And I think one of the things that unites me compared to the others is that no one has this reporter mouse line that we've generated that enables us to very efficiently purify those cell populations. And if we were to rely on the old lines, I don't think that many of the things I've proposed would be feasible.
Okay. Dr. Srinivas, please.
Hi, thank you for your presentation, and you've already had a lot of questions. So let me start with a few that are a little bit on the administrative side. So you have a 60% time commitment. How will you ensure adequate oversight of all three of these aims with a 60% time commitment?
So I do have also some other grants, but I can assure you that a big portion of my lab will be devoted to that. I plan to have at least 2 PhD students working on each of those 3 aims, and all of that will be done in parallel. I'm planning to have weekly meetings with those students to make sure that they all progress in good pace. And I think it should be feasible to oversee all of the aims together.
Another question, have you considered any, or are there any ethical considerations that arise from creating human gonadal organoids? And how can you address these?
So for now, we're not yet planning to generate human gonadal organoids. We're only planning to generate mouse gonadal organoids, but as a follow-up study that won't be part of this ERC, but maybe part of future ERC or ERC proof of concept, we are planning to move to human, and once we will get to that, we will get all of the ethical improvements that will need to be in place to do that.
Right,
Yeah.
but I asked about ethical considerations. So is there anything you should perhaps take into account before generating human organoids like this?
So it's kind of outside of this grant proposal, but I will reply anyway. So there are all kinds of groups of patients that us generating testis organoids for them, and hopefully those testis organoids be functional, will be extremely useful. I'm mostly talking about patients who are infertile, who are not able to have a biological child and are very keen of being able to generate some sperm. So the hope for those patients is that once we'll have the mouse testis organoids platform, we will be able to convert the protocols of what we do with the mouse with human cells, but mostly working with human iPS cells that we will be able to generate to differentiate towards PGCs, the primordial germ cells, and also the somatic cells, and then to construct fully artificial testis organoids. Of course, if we were to generate sperm there, we won't be able to test it functionally because we won't use it until we know it's sure, but I think we still have some years until we'll get there. Another group of patients that it will be extremely relevant to are prepubertal kids that undergo cancer therapy, because when someone gets chemotherapy, a side effect of the chemotherapy is that it creates, in many of the cases, infertility because it damages the spermatogonial stem cells in the testes. And whereas adult males can freeze a sperm biopsy before chemotherapy, kids cannot do so, and kids until the age of 13 do not produce sperm. So nowadays, when a child has cancer and it needs to undergo chemotherapy, we don't have any solution in the medical world to allow him to preserve his fertility, and that means that he will survive to adulthood, but very likely remain infertile. So this is also a group of patients that we believe that our testis organoids will be of use for. I won't say it because it's not part of but we have now a proof of concept grant to do that, but this didn't happen when I submitted this grant.
Maybe,
Thank
yeah,
you.
do you have more? Do you have more questions?
Maybe a quick question, and if you can be brief in your response. You will generate a large amount of multi-omics data from this project. How will you distinguish causality from correlation from all these data sets, right? And then also, will you make this openly available?
Yes, so of course, as part of the ERC, everything is openly available, and we will upload all of our data to be fully accessible, including the original sequencing data and the analyzed files of sequencing. So that's about the openingness. About the causality and correlation, this is hard to distinguish, but we will only choose to model candidate enhancers once we'll pile up a lot of positive hints that make them a more promising candidate. If we're just going to see something open in 2 or 3 stages, that will not be something that we will immediately choose to model. And also, until we'll have our organoid system up and running to be able to screen in it, since we plan to first screen everything in vivo, we will have to be very picky with the candidates we choose to screen.
Okay, maybe Professor Jacoli, you want to ask question, maybe one question, and then we start with the discussion afterward.
Yeah, thanks very much. So just quickly, if you could briefly summarize your thoughts and your plans for data management. It's a lot of data. You mentioned making them available, but do you use version control, or which softwares or which platforms, servers, if you can tell us about that?
Yeah. So yes, so my lab, all of my lab's data, including the sequencing and all of the data that we generate, is being stored on a a Dropbox account that is a kind of commercial Dropbox account that we pay, and everything is being backed up in case there will be some cyber attack. Once we publish things, we upload everything both to the GEO and also to other respiratory deposits that makes the data available. In addition to that, a lot of the analysis, the computational analysis, is being done on our lab server that I'm planning to buy as part of the budget, the extra budget that I'm getting from the ERC. And all of my computational students will analyze the data there. And I think that's it. I mean, many labs nowadays generate a lot of multi-ohm data and single cell data, and it's very kind of pipelined of how to store them and how to make them openly accessible.
Right, but so Dropbox servers are in California. So do you envisage to put any patient data, confidential data on servers in California, or how do you deal with that?
No, so I don't have the patient, like I said, my lab is only generating mouse data. I don't have the patient data on my servers. The only thing that I will be getting from the patient data is VCF files of the variants that were found in them. I won't get first of all, I just have an identifier. I don't know the identity of the person, and I don't have the raw sequencing data. That is being stored in my collaborators, and they are all also clinically managed, so they have their own ways of making it not accessible. The patient's data is not mine, and it's not going to be accessible through me.
Thank you.
Okay, a little brief. Yeah.
Okay.
I think you remembered.
I could answer much better because much of the things have been published, and I know at the time I don't think I knew a lot of the stuff that you are asking now.
Yeah. Thank you very much, Dr. Ghanen. I would like to ask you, what was your feeling during your interview, like your real interview, and today? Just this is like audience that they want to know, how was it today and also the real one?
So I saw that today took me 1 minute more than it should have been.
Yeah.
But at the time it didn't, because today I just opened 2 hours ago my notes to try to remember what I said on what. But back then I was really on the time. So I think this is really important to practice. And I really had everything written on the side on exactly what I want to say on each slide, because it's very easy. I mean, I think that as a scientist, we are not used to being such time strict, and it's very tempting to sometimes add a few more words here and there, but it's really important, if you want to stick on time, to not be dragged to those places. So I feel that now, but again, I'm like 3 years into my ERC, I'm much more mature and experienced than I was, and I know to answer much more I don't know, with confidence on some things that I know that we've already succeeded and published than I did then. But overall, my experience in the interview was also really good. Something that I was told when I was doing a lot of mock interviews is that someone who was interviewing me, that was actually from physics, she was hearing my talk, and then she was asking the questions, and she told me, it's like 2 different complete different people in the talk and in the questions. Like in the talk, you talk like a bit like a robot, because you've tried that for so many times and it's very like automatic. And then in the questions, it's like you're all lively and up and happy and engaged. So I think this is kind of a tip to others to try to. I know that it's very stressful, and I know that by the time you do the interview, you've tried that maybe 50 times, but it's really important to try to be yourself also throughout the interview itself, and not just in the questions.
Now I would ask panel members, how do you perceive when Dr. Ghanen answered the question? Do you think it's sufficient? Do you think it's too much? Do you think that she can do this and this better, or was sufficient and perfectly perceived? No comment on that? Yes, Dr. Professor.
Yeah, I think you did really well. Obviously, it's a bit of a strange position to be in, given that we know you've already got it. if anything, perhaps you could have been a little bit briefer in your responses, because I think some of the responses were very detailed. And of course, like you said, it's different because you've already done some of this. So you could hear that confidence, which I think you would not have because you haven't done it yet at an earlier stage. So I think the only thing I would change is maybe be a little bit briefer in your responses.
I mean,
Professor.
if I could just add something to that. So it didn't happen in my panel, and maybe it didn't happen to you either, Nitsan, but I was warned a lot actually that it can be that that panel members will just cut you off. And they're doing this and just for the other people, just so you know, and because this can be very demoralizing, I think, if it happens in the moment. So they're only doing it because they're trying to get through a list of questions that they have in the 18 minutes. And so it's in your interest then to be short enough that they don't feel compelled to do that, because I don't think it's very pleasant for them also. But if it does happen, don't get trapped in thinking about that. Just just move on and answer the next one quickly. But I just to add that.
No, that's good point.
Something that
Yes.
I was advised when I was interviewing is that to kind of collect all of the questions that everyone in the mock interviews ask me, and all of my worst questions, like you're not a computational, how will you deal with that? And try to write to yourself short and precise answers. So you won't so first of all, it won't be too long, and that you won't drag into places, because sometimes we are our worst enemy, or trying to raise up all of the problems ourselves. So to not get into that points. So try to prepare yourself, and like Gutam said, I think that it's really important to kind of make the answers short. So which I'm not sure I've done here, but in order to make them ample of time to ask you all the questions they wanted or they need to ask you.
Professor Jekyll.
So obviously, yeah, you were very good in answering, because of course you went through this. But I agree that a good panel has to be tough, and you have to kind of feel the stress and be prepared for that. For me, one of my most stressful moments was the ERC interview. It can really be stressful, and I think it's good to select the kind of tough colleagues for that, who really try to go and take apart your proposal.
Okay, I agree with that. One thing that I consider, and I would also people want to go to this step of the interview, is that I like this. You always smile and like give positive vibe. I like it, because that's also important, I think. But I agree with all that was relying. Of course, you didn't prepare. That's definitely that time you will prepare to make it very well. And also, panel would like to evaluate you, have all answer of the question that your referee asked, or they raise the question for them to evaluate it. This right, and they have this, like, I don't know, 20 question, then you have 18 minutes, meaning that, I don't know, each minute question and answer, each minute question and answer, yeah? So what would you suggest? One of you say that, what would you suggest? How many sentences should have an answer? 3, 4?
It's probably better not to make a rule like this, because
Yeah.
there will be some question, like maybe the entire thing is hanging on this one thing that the whole panel agrees they're stressed about, and then it's okay if it takes 5 minutes to discuss that. So I think you just have to.
I think that 5 minutes is really long to
Yeah,
have.
no. I was I mean, that was an exaggeration. I guess what I would personally advise against, like, setting a rule, like you have to have 3 sentence answers or something,
No,
because
I mean just like to have a kind of idea for people.
I think an answer per minute is basically what you would.
Exactly. Yeah.
But
So
I think if you manage to think in advance, or people ask you in advance a lot of questions, then you can think of them when you are calm and not in stress, and write down the answer to that and read it a few minutes before your interview, so everything will be fresh in your mind, and then not be dragged to a too long question.
Yeah, and I think this tends to be especially useful for things like that are very open-ended or even a little bit philosophical. I always get asked, like, why should we care about evolution? And of course this is like, I don't know, we can talk about this for hours, right? But so for that kind of thing, it's almost impossible to come up with a good response on the spot. And so those kind in everyone's field, you will have some questions like this, which people like to just ask, and then you just have to.
Any question from audience? The people would like, they can write in the chat. One point that I like it, you did it, and also everybody who is science should do it. You bring fact, side, things that I like it a lot. Like you said that this lab, this team, I know, that's showing that you're updated about your field and answer the question by bringing correct, valid science. I would also recommend to do that, and I did the same actually. Actually, I have one technical question, and really I said that, okay, one lab established this methodology, of course, for different model system. I will use this for mine. Are the interview always in person, or are they sometime online?
Starting run is only online these days.
Yeah,
The
mine
panel,
was online.
yeah, the panel is together in a room in Brussels, but you will be online.
Yeah, I think that basically ERC updated after COVID, or from COVID till now, all interview
Only
hold in
synergy, only synergy is.
only synergy in person. That's I didn't know. Okay, good to know.
Yeah, the synergy is in person and the rest are online. And another thing to say is that they're using a very annoying platform, which is not Zoom. So I very much recommend that you try that platform in advance and make sure you know how to share it and how to operate it. So it won't be the interview day being your first day trying to operate that system. I forgot how it's called, but it's not a friendly one.
WebEx.
That
Yeah,
one.
WebEx, yeah. How many minutes should a presentation last for ERC StG? Depending of the panel, basically. Each panel write you the time and then the instruction. What weight has the interview on final evaluation order? This is a
100%.
question?
I mean, it's unlike some other evaluation schemes. Once you're passed to the interview, like the panel has access to a scored list, but then in the end they decide how many to award or recommend for funding. So it's like there's no, yeah, it's not a weight. It's like it's just a next step.
No, but I mean, I don't know how, I mean, I also don't know the answer to that, but can the interview really make a big difference in the order of people within?
So apparently, I don't know if Gaspar or someone has been on the panel already. What I've heard from people on the panel is that it only really makes a difference for the people who are close to the cutoff. The ones who are already very low, it's hard to pull them up, and the ones who are very good, they are already in a good position.
Exactly. No, I agree. That's it. Just all depending of the external referees evaluation, and the interview really determine gray rank actually, who may cut off. Yeah. Is all question or topic related, or do they ask why you kind of question? So for me, it was pure scientific question, but I heard that they ask out of the science topic question. Who has this from panel?
I had mostly science ones, but they asked me, for example, about the budget because I was asking some extra money for a transgenic facility. So they were asking me, why do I need this extra money and why this is not part of my setup package and so on. I didn't know if it's a good sign or a bad sign, but they asked me some other stuff.
Any other comment on that? Okay.
I mean, I don't know if it's in the category, and it kind of got asked today in the mock, but they can ask about competitors, postdoc advisor, separation, like all these kind of things. I don't know if that would fit the YU category, but they
No,
will.
but I also heard that they can ask, I mean, not me, but they can ask, why are you different than your postdoc mentor? How do you know he's not going to compete with you and do the same things? So this is things you need to think of what makes you, what defines your niche, or what makes you unique, or even just saying that my postdoc mentor knows about what I'm planning to do and he guaranteed that he's not going to go into that path is a good enough answer.
There's another question. I'm writing few tasks that are not my expertise. If they ask how I will do them, can I basically just say I will hire experts.
This is
Well,
what they asked me about the non-computational. I think that was my biggest fear. I had a question in my question thing. You're not computational, because I was also applying for system biology, which is LS2, a lot of the multi-omic stuff, and I don't know to write a single code sentence. So I was terrified about that question, and I was asked about it here. So I've been trying to prepare myself with some answers, but yeah, you know, the imposter syndrome will always be there, and you're never going to be sure that you can do them. But I think it's very valid to say that you will hire some experts in the field, and this is what I said, that
yeah.
to say that
I
first
would
of all, it's not such a big deal. Many people do it. It's not like I'm developing a pipeline myself, but secondly, that there are many people who know how to do it, and you will hire them.
Yeah, you could also use that moment to call out specific future or present colleagues in your institution or expertise in your institution, because one of the things that gets assessed at various points is like how well you fit also, and how much support they're going to give you. So this could be a way to, you know.
Fit it in.
Yeah, but also is, I think, weighting also, like how many tasks. If you write a lot of tasks that, okay, I don't have expertise, I don't think doesn't make sense. Like, but in your case, you have most of the tasks. You can do just one. I think it does make sense. But if you have many tasks, then they said, what do you want to do? Either your collaborator will do, or I don't know, you should hire a scientist that has expertise, but you cannot supervise even this person because you don't have yourself this you know, that's really kind of balancing, I think. How many external reviewer are needed? 2. What if the reviewer are contradictory? Okay, 1A, second C. Are the reviewers chosen randomly, nominated by panelists, or is that typical? Is there a permanent group of external reviewer? So many questions. Okay. For me was 7 common, and like 7 people that evaluated me, but I heard that also some people 6.
I had 11.
Okay, see 11, but for me it's 7. I think they are different. Which LS did
I
you?
had 8.
8, okay. I was LS5. And then
So the external reviewers are supposed to be domain experts, and so it's not a fixed group. There
no.
they will try to really find people that really know what you're talking about. But then there's lots of exclusion criteria for conflict of interest. So in small fields, it can actually be difficult for them to find enough people. But
Exactly. There is no fix, but they have a list of the, you know, for different area they have, and then also like ERC award is coming, and then also they are expert in the field. They will ask them also in the future. They usually invited 5 people panel nominates, maybe 11 to 12 will accept. LS3.
that was me.
Yeah, you answered. I'm sorry. Yeah, I was thinking. Yeah. Good. I think that we are running a bit now after the time point. So at this point, I would like to thank, really, it was really fantastic, I think, session. And thank you, of course, our ERC awardee, our speaker and panelists for involving and discussion. And thanks all contributor and all people that hear us and also ask the question. Good. If
Thank
you
you
don't
all.
have
It was
any,
much nicer than the real one.
definitely. So thank you very much. Have a nice afternoon. And if you have any question, you may contact the organizer of the workshop, and also will be in the YouTube of the YouTube channel of the Young Academic Europe soon. Have a nice afternoon. Bye.
Bye. Thank you.